Okay, thanks for inviting me to be part of the tutorial. It's a real honor to be here. Um, Today, I'm going to talk about some of the results that we've obtained over the last, 10 or 15 years working on the evolution of the G matrix. The idea is to give you some intuition for how the G matrix responds to various evolutionary processes, right? And the work I'm going to talk about is a collaboration that started when I was a postdoc in Steve's lab around the year 2000, so it's been a while now. And we did this work with Reinhard Berger. And so this work has kind of continued sort of in the background, as we've done all of our various other things. But we've continued to, gradually make progress. And I'm going to talk about some of the stuff that we've learned along the way. All right, so Steve already talked about the multivariate Breeder's equation and the reason that we might be interested in the evolution of the G matrix per se. The reason, obviously, is that the G matrix plays a central role in predicting the response to selection, right? It's one of the most important evolutionary parameters. And you already know that beta is the vector of directional selection gradients, and G is the genetic variance covariance matrix, the additive genetic variance covariance matrix. You can also use this equation by rearranging it to reconstruct the history of selection, so you can calculate net selection that's happened in any system if you know the change in the phenotype over time, and you can assume that the G matrix remains relatively constant. Of course, you might also want to predict what's going to happen in the future, and you can use this response to selection equation to do that as well. All right, but like Steve said, for quantitative genetics theory to be valid, the estimate of G has to be representative of G for the time period you're interested in, right? So G has to be relatively stable for whatever period of time you're interested in talking about. And that sort of motivated the work that we did on the G matrix. And the question was, how often and what circumstances result in a G matrix that's stable enough that you can actually infer things about the population using it. All right, and so this was sort of the situation back then, around 2000, when we started this work. So people had done empirical comparisons of G between populations within a species. Usually those produced fairly similar G matrices between different populations within a species. If you looked at G matrices at higher taxonomic levels, you saw less stability, which kind of makes sense. If you go to high enough taxonomic levels, then of course the comparison doesn't even make any sense, right? Because you may not even have the same traits anymore in different taxa, right? So at some level it definitely changes, right? At some level you're even dealing with different sets of traits, and so you can't even make the comparison. The analytical theory, basically the state of that was that on the one hand, it couldn't guarantee G matrix stability, but on the other hand, we couldn't really predict how unstable G matrices would be on the basis of analytical theory. And we're sort of in that situation now. And so an obvious approach was to use a simulation approach. At the time, around 2000, people had already used computer models to study the maintenance of quantitative genetic variation in the univariate case, but nobody had really done much in a multivariate context, right? So if we were interested in the G matrix and genetic constraints imposed by the G matrix, nobody had really taken a simulation approach to address those problems at the time. And so our goal was to use individual-based models to look at the stability of G over long periods of evolution. More recently, we've become more interested in the evolution of the M matrix, which I'll talk about in a few minutes. And even more recently than that, we've gotten interested in how epistasis can affect all these different phenomena that we're talking about. And I'll talk a little bit about that at the end, right? But it gets a little bit confusing. I'll try to keep it simple. All right, so for the tutorial, I think it's worthwhile to go through some of the methods that we used, because maybe in your own research you might want to do some of this stuff eventually. So basically, these are Monte Carlo simulations where we keep track of every gene and every individual in a population. If you have any questions as I'm going along, feel free to interrupt me, because we have 30 minutes of discussion. If we could use it up along the way, that'd be fine too, because then we could meet our goal of never taking breaks. So in this model that I'm going to talk about, and actually all of our models, we assume that all loci are pleiotropic. Okay, so every locus, and we assume 2 traits, right? So we haven't dealt with more traits, at least not officially. We've done some unofficial models involving more traits, and the results, for the most part, are similar. For the first model I'm going to talk about, we assume additive inheritance with no dominance or epistasis. Allelic effects are drawn from a bivariate normal distribution with means of 0. So it has to be a bivariate normal distribution because the loci are pleiotropic, so every locus, every allele has an effect on both traits. Does that make sense? So each allele has 2 allelic effects, one for trait 1 and one for trait 2. These kind of details are important to get early on because otherwise things won't make sense as we go along. And so that means there is a matrix that describes this bivariate normal distribution. So you have 2 variances and a covariance that describe the distribution of new allelic effects that enter the population. Does that make sense? That's actually the M matrix that I'll talk about in a few minutes. There's a mutation rate. We use a relatively high mutation rate so that we can actually maintain genetic variation in these populations. For a reasonable population size, if you use realistic mutation rates, you don't maintain realistic levels of genetic variation. Why that is the case is still a little bit of a mystery, and so that's something that people could try to figure out if they feel inclined to do that. This model also involves environmental effects, and we draw those also from a bivariate normal distribution with a mean of 0 and a variance of 1. And so in a way, this kind of sets the scale for the model, because if you think about heritabilities, it's the additive genetic variance divided by the total phenotypic variance. Well, this is kind of, determining the amount of phenotypic variance that you have. And so in some ways, that's kind of like the scale of the model. All right, we assume an individual selection surface that's also shaped like a normal distribution, and we usually use strengths of selection of 9 or 49. I'll tell you what omega means in a minute. Each simulation run has a period during which, about 10,000 generations normally, during which the simulation reaches kind of a quasi-equilibrium between mutation, genetic drift, and selection. And then after that, we have experimental generations where we keep track of the variables that we're interested in. Okay? All right, this is the life cycle that we use. So basically, we start with a population of some number of adults. N is the population size, or it's the carrying capacity. In these simulations, the population always remains at the carrying capacity, and it can never go above the carrying capacity, as you'll see in a second. So those individuals produce progeny. Usually the mating system is monogamous, unless we have some reason to do a different kind of mating system. We just assume regular Mendelian assortment and a sexually reproducing organism, right? So that it's easy to, keep track of whose alleles go to which individual. Each individual obviously inherits one allele at each locus from each parent. We have mutation and recombination during this phase of the life cycle. Then we have some number of progeny that are produced, right, where B is the fecundity of females, basically. And then we have selection. They're going to be greater than N survivors under most of the parameter combinations that we use. If they're less than N survivors, then we terminate that run because we don't want fluctuations in population size to affect the results. If they're greater than N survivors, then it continues, and we only allow N individuals to be to make up the next generation, right? So any individuals beyond N, individuals are randomly culled until we get down to that carrying capacity of N. All right, usually we start most runs with a population of genetically identical adults. And like I said, we have those 10,000 generations. We impose some movement of the optimum, right? So where you have a selection surface, and maybe it's stable, maybe it's just stabilizing selection with a stationary optimum. Maybe it moves stochastically. Maybe it moves in a directional way. That's something we can impose in the model. Like I said, we calculate the G over several thousand generations where we keep track of variables, and we usually focus on average single generation changes in G because we're interested in how things affect the stability of G, which can be measured as a single generation change in some aspect of G, like the variance or the covariance or the angle, something like that. Okay, so like I said, the M matrix is really important, it turns out. for the genetic architecture in these systems. And so you can think about it this way. You can draw an ellipse which sort of describes this bivariate distribution of new allelic effects, right? And so like I said, we have trait 1 and we have trait 2. In this M matrix, the mutational variance is 0.05, right? So I'm going to talk about the mutational variance as we go along. This is just the variance in those allelic effects for trait 1 or trait 2, right? So what it means is if you determine that there's a new mutation occurring at a particular allele, you just draw a random number from a distribution described by these parameters and add it to the existing effects of that allele to get the new allelic effects. Does that make sense? So if there's, yeah. Okay, so when there's a mutation, that means the allele is going to change. So you have to change it in some way. And the way we change it is we draw a random number that has a bivariate normal distribution from a distribution described by this matrix, right? So the variances are on the diagonal and the covariance is on the off diagonal. Right, so if this correlation, if this covariance is 0, then what happens is the effects on the two different traits are independent, right? So there's no correlation between those effects. And so you'd get something like this. So if I get a large effect, say for trait 1 of that mutation, then I could get any effect on trait 2 because those effects are going to be independent. They're still inherited together though, because that allele affects both traits, right? Because the loci are pleiotropic. All right, it's a little bit confusing, but it's really, really, really important. It's the most important. This is one of the most important slides. The important ones should be at the end probably, huh? But all right. So now imagine though that there is a correlation, a mutational correlation, what I'm going to call a mutational correlation, okay? Now it means that the mutational effects are no longer independent, right? Instead they're correlated. So a mutation that tends to increase trait 1, when you have a mutation that increases trait 1, you're going to tend to get allelic effects that also increase trait 2, right? So this could happen if you have, for example, two traits that share the same developmental pathway, right? So if you're talking about, I mean, in a really sort of obvious example, if you're talking about a symmetric organism and you're talking about right arm length versus left arm length, a mutation that increases my right arm is almost always going to increase my left arm, right? Because they're determined by the same developmental genes for the most part. So those might have a very strong mutational correlation, right? The thing you're going to have to get used to is that you can have pleiotropic effects, right? So every locus can be pleiotropic, but it doesn't necessarily imply that there's a genetic correlation between the traits that are affected by the pleiotropic loci, right? Most people think pleiotropy means there's a genetic correlation. Actually, you'll see from these models, we always have every single locus is pleiotropic. The most common result is to get no real genetic correlation, right? Because pleiotropy only produces a genetic correlation if somehow the allelic effects are correlated at individual loci. Does that make any sense? You'll see more as we go along. I feel like I've completely lost everybody. Okay, so for this slide,
Thursday in one of my talks, I'll use a simulation program as a similar model to this, but what in that program it does is that a mutation increases or decreases the output of a gene, and that gene then has some coefficient of how it contributes to the trait. So I think that would correspond to R equal 1.0 here, either with plus or minus 1.0, because you're increasing the output of a gene or decreasing it, and then there's just a multiplier, positive or negative, that says how it contributes. There's one for each trait.
Yes.
So I'm just trying to relate it to this.
Something like this.
It would be like that, but much
even tighter correlation.
Even tighter.
Yeah. All right. So to summarize this slide, this just describes the new mutations that enter the population whenever there's a mutation, right? So basically you draw one of these points and you just add it to the existing allelic effects to get the new allele. That's all it is, right? And this is just a circle around a bivariate normal distribution. So it's actually really simple. So you just draw a random number from a bivariate normal distribution. Then you get two effects, trait 1 and trait 2. You add them to the existing effects, and that's your new allelic effect. And then those allelic effects are always inherited together for a particular allele. Yeah.
You had an incredibly incompetent draftsman for this figure, I noticed. And the means, the position of the mean, is the scale for that isn't indicated. Is it 0?
Yeah, it's 0. So we have no directionality to this, right? So we assume that there's the mean effect of mutations is just 0. They can be positive or negative, and they're equally likely to be positive or negative. If it weren't 0, then you would just get mutational drive that would drive the population to new phenotypic places in phenotypic space without any selection or anything else. Mutation by itself would drive things, would drive the evolution of the mean phenotype. Okay, then the other thing, there are only 2 of these slides that are these concepts that you really need to understand. So the other thing is the selection surface, right? So the other thing we impose is an individual selection surface. So this is very much like what Steve was talking about. You know, it's the adaptive landscape or the individual selection surface. So this is going to be described by omega, which is a matrix, which is related to gamma. Right? So now we're switching, so this is the problem, that we're always switching notation, right? So now we're switching from gamma to omega. Omega also describes the shape of a bivariate Gaussian surface though, right? And so what it's doing is the diagonal elements are basically variances, right? Describing the variance of a bivariate Gaussian surface. And so smaller values are a smaller variance, so it makes for a steeper hill, right? Larger values are a larger variance, so it's a flatter hill. So small values indicate strong selection, and large values indicate weak selection. And then if you have any non-zero off-diagonal elements, then that's analogous to a covariance, and so that's going to indicate correlational selection, right? So when the off-diagonal elements are non-zero, then you'll get something like this, like a ridge of selection. And so under these circumstances, individuals that have certain trait combinations are going to have higher fitness than individuals with other trait combinations, right? So the combination of traits matter. There's an interaction with respect to fitness. Over here, the traits sort of independently affect fitness, right? And so we're going to care about both of these things because both of these affect the G matrix. All right, and then beta is basically going to, if the population mean is somewhere on here, beta is basically going to point toward the optimum, the bivariate optimum. Beta, the selection, directional selection gradient. Does that make sense? Okay. And then the G matrix you've already been introduced to, so I'm not going to spend any time on that. But, again, you can draw it as an ellipse. And that's described by the genetic variances and the genetic covariance, or it can be described by the leading eigenvector and the eigenvalues. And we're only going to deal with the 2-trait case, so basically we only have to worry about the leading eigenvector because we know the other eigenvector is just orthogonal to that leading eigenvector. Yeah.
So I was just wondering how omega relates to beta, because I was thinking when on your previous slide, when you have the non-zero of diagonal elements on omega, that's sort of that would be. That would mean that beta is pointing towards either increasing or decreasing both traits simultaneously. Am I right, or am I completely confused?
No, beta still points toward the bivariate optimum, but the relative strength of selection on the traits would be different than in a case for the same location. It'd be different than the case of a perfectly circular individual selection surface. But the way to think about it is, the easiest way to think about it is the more of these lines you have to cross, the further away you are from the optimum in terms of fitness, right? So in this model, if you're right here,
Okay.
beta would be relatively weak compared to being right here, right? Because here, you're not that far off. It's a ridge, so you've gone down a little bit from the peak. If you move this direction, you're just kind of gradually moving away from the peak in terms of elevation, right? So think of this as like a topographical map where this is like the peak of a hill. If you're moving this direction, you're moving very slowly downward in elevation. But if you move in this direction, you're moving very rapidly down. And so there's much, much stronger directional selection if you're down here than if you're over here, right? But the directional selection is still toward that bivariate optimum.
Could I quibble with a statement? I think in the right-hand case, the arrow does not point directly towards the optimum, but it will point towards the ridge. If you're off the ridge.
So if you're
Say
here,
there. If
you
you're
think
there.
beta will point.
For example, if it goes straight up contour there, it'll actually not be pointing directly to the optimum. It'll be at an angle.
I don't. Yeah, that's right. Yeah, that is true. Yeah,
So
yeah.
if you have a long, thin
No,
ridge,
you're right
you'll be
about that.
tending to climb the
Yeah,
ridge
you're right.
first and then
Yeah, yeah.
turn and go
Yeah.
along the top of the ridge.
Yeah, that's correct. Yeah. That's the easiest way to think about it, is what Steve just said. It's the direction of steepest uphill slope, right? So if you're on a hill, you face toward the steepest part of the hill in your local vicinity, and that's the direction beta points. Yeah, that's right.
Or if you're a skier, you would point your skis towards the fall line, right? But in this landscape, you point them at 180 degrees from that. If you're a skier,
Except you don't normally just ski straight.
I retract
That is the fastest way to get to the bottom, though. Okay, and so what we're going to care about mainly with respect to the stability of the G matrix, we're going to care about the stability of the various elements of the G matrix, but we're also going to care about the angle of the G matrix, right? And the reason that we care about the angle of the G matrix is that as far as the evolution of genetic variances, that's relatively easy to understand in the univariate case. But when we start talking about multivariate evolution, we also care about genetic constraints. And the genetic constraints are imposed mainly by the angle of the G matrix, right? So if its angle is pointing a particular direction, and it's a different direction than the direction that beta is pointing, then the G matrix will be a constraint for the response to selection, right? And so we're interested in the angle, at least we were most interested in the angle of G, because we see that as an evolutionary constraint. All right, and so now I'm just going to go through some of the results that we found, to kind of give you some intuition on the evolution of the G matrix, right? So here's just a plot over generations of the angle of G, right? So it can be from, and this is just in bivariate trait space, right? So you can imagine that it's just a plot of trait values. And so it can be from minus 90, which is pointing straight down, to plus 90, which is pointing straight up. A value of 0 means it's pointing along the trait 1 axis, right? And so a value of 90 is pointing along the trait 2 axis in phenotypic space. All right, and so you can see with no selectional correlation and no mutational correlation, there's quite a bit of evolution of the angle of G. It's just kind of going all over the place. But there are periods of relative stability. It's not just a random draw every single generation, right? There's some history here. But then you can reach areas where it's incredibly unstable for some period of time, right? And so this is sort of just kind of baseline data showing you just the stability of G without a lot of constraints on the evolution of G. So what can actually stabilize the G matrix? So one thing that can stabilize it is stronger correlational selection. And so if you increase the correlational selection, so now evolution goes from being on a perfectly round hill to being on a ridge, then that will stabilize it, right? So now we get so with a selectional correlation of 0.75, we get a considerably more stable G than we had in the case with no correlational selection. All right, and there's just a table here showing the relative values. That doesn't really matter. If we impose a mutational correlation, that can also stabilize G, right? So if mutational effects tend to be correlated so that if you get a positive effect on trait 1, you also get a positive effect on trait 2, then you tend to get a very strong genetic correlation because that mutational correlation directly, those mutations directly affect the standing genetic variance, the additive genetic variance, and you can get a very strong genetic correlation just due to these mutational attributes of the system. And so here's an example. So in fact, under these parameter values, the mutational correlation is actually much more effective at stabilizing G than selection was. Yeah.
What are those pieces of the schmutz at the bottom there?
Those are just slight changes from this value. So if it just goes, so a 91 degree angle is actually a negative 89 degree angle.
Oh, I see how it's working. Okay,
yeah.
Got it.
Because there's no directionality to which direction it points. All right, if you have both a positive correlational selection and a positive mutational correlation, then those two things reinforce each other and you get an even more stable G matrix, right? So if there's a positive mutational correlation and positive correlational selection, then you get a very stable G matrix. And in really extreme cases with very strong correlational selection and very strong mutational correlations, it basically just locks in at a 45 degree angle for an entire simulation run and just stays there. But if those forces are misaligned, then that actually destabilizes the G matrix, right? So if they're opposite signs, then that actually decreases G matrix stability. And so the easiest way to see this from this graph, different lines on this graph represent different mutational correlations. So this line right here is a mutational correlation of 0. The x-axis shows the strength of correlational selection. So on the ends, you have either strong positive or negative correlational selection. This is unstable, right? Because there's a larger change in the angle of the G matrix, so higher values are less stable. And so you can see that for this graph with no mutational correlation, increasing correlational selection, it doesn't matter the sign. If you have increased correlational selection, you get a more stable G matrix, right? And that's kind of a symmetrical relationship. But now look at this line right here compared to that. So here there's a mutational correlation of 0.25, so it's not that strong of a mutational correlation. If it aligns with correlational selection, then it actually increases G matrix stability. But if it's misaligned, it results in a huge destabilizing effect on the G matrix, right? So misalignment of the mutational correlation and the selectional correlation actually decrease G matrix stability. All right, a few other results. A larger population has a more stable G matrix. So most of the fluctuations in the G matrix are probably caused by genetic drift. And so if you increase the population size, those fluctuations get smaller because the effects of drift are smaller. Asymmetrical selection intensities or mutational variances can also produce stability, even if there are no mutational correlations or correlational selection. And the reason is that then you get an elongate G matrix in one or the other trait dimensions, and eccentric G matrices like that tend to be more stable than round ones. All right, so this just shows the effects of different mutational variances. So as they get more different from one another, you get more stable G matrices, and if there's really asymmetric selection pressures, then you get a more stable G matrix than if they're not asymmetric. Yeah.
So just like talking from a geometric point of view, not necessarily that mathy, but when you said that the more like round, less eccentric matrices are more prone to be disturbed, then you won't see that change, right? Because I mean, if I'm imagining the extreme of a circular matrix, then it doesn't matter how much it rotates, the eigenvectors won't change, right?
They'll change, their orientation will change, but it'll have very little effect on the response to selection, right?
Okay,
So that's
yeah,
actually
okay.
the good news. This is part of the good, that's one of the good news points that I'm going to get to later, is that round G matrices tend to be incredibly unstable if they're perfectly round. But they're not really an evolutionary constraint anyway. So it doesn't matter how unstable they are because they're barely constraining anything. When they get very eccentric, then they're a major evolutionary constraint, right? Because they're really going to constrain the response to selection. And those actually tend to be the most stable G matrices. So when you have genetic constraints that are important, those are going to tend to hang around for a long time, at least given the assumptions of our model. If you have genetic constraints that are unimportant, they'll change a lot, but it doesn't make any difference because they're not really constraining anything anyway. The beta is driving the direction of selection more than G. And so that's a really good point, and it's really important. But it's encouraging because it means if you find a very strong genetic constraint in the system, that will probably hang around long enough that you can make some meaningful predictions using it. All right, so these are the conclusions from this first paper on the stationary optimum. Correlational selection increases G matrix stability, but not very efficiently unless selection is very strong. So it takes very strong correlational selection. Oh, do you have a question?
Here. So on the last slide, why is it that asymmetrical selection intensities or mutational variances produce stability? Functionally, what's actually happening? I just didn't understand the last statement.
Oh, the reason is that it makes it more eccentric.
Okay. Oh.
And those eccentric G matrices just tend to be stable.
Oh, but okay.
Yeah, and
Okay.
so the reason it makes it more eccentric is if you have strong selection on one trait and weak on the other trait, then it decreases the variance in one trait and allows more variance in the other trait. And the mutational variance has the same effect.
So my question is, why inherently is a more eccentric G matrix more stable? Is it just because there's not as much variation that's off the sort of primary axis? You can't sort of be pushed in weird directions. You sort of all the variation is along one axis.
I think so. Yeah, I think that's the main effect. And it takes larger effects due to genetic drift, larger changes in allele frequencies to change the orientation of a really eccentric G matrix compared to a round one. Because very small changes in the variance of either trait change the orientation of the eigenvectors in a round G matrix, but in a really eccentric G matrix, it takes a lot more to change the orientation of the eigenvectors. Are there any? So I'll just go through these conclusions and then see if there are any other questions. All right, so the main things that cause eccentricity and stability in a G matrix are correlational selection and mutational correlations. Correlational selection has limits because it has to kill individuals off, right? There's a cost of selection involved in maintaining G matrix stability if you do it through selection. Mutational correlations are very good at maintaining stability without that cost because they directly affect the standing genetic variation in the population, right? So they have sort of infinite potential to increase stability. G matrices are more stable in larger populations or with asymmetries and trait variances that can be caused by mutation or selection. If the mutation and selection matrices are aligned, then you get a more stable G matrix. The eye-opening thing for us was how important mutational correlations were. We weren't really going into this really thinking that much about the importance of the M matrix per se. And so after the first couple of studies on G matrix stability, we realized that the M matrix was a very, very important thing that we needed to understand in more detail. And the take-home message from this first part of the talk is that for some suites of characters, the G matrix is probably very stable. So those things that are highly integrated or that traits that work together, like upper mandible length and lower mandible length, something like that, or things that share developmental pathways, those are probably going to have very stable G matrices. But traits that aren't integrated in those ways are probably going to be the ones that have the less stable G matrices. And so different types of characters, depending on whether they're functionally related or developmentally related, those are going to be the ones that are going to have the most stable G matrices probably, and those that are more independent from a morphological standpoint or developmental standpoint are probably going to have the less stable ones. All right, but like I said, if you see a very strong genetic correlation, those types of things based on our models at least are the kinds of things that would persist for a long time over evolutionary time. All right, so what happens now when the optimum moves? So each one of these parts is going to get a little bit shorter than the previous one, so don't worry, I'm not going to be going on and on forever. So we can ask, okay, so the first set of results that I talked about were just a stationary peak, right? So it's just evolution with stabilizing selection. The peak doesn't move, it just sits there. Okay, now the question is, what happens when the peak moves? Okay, so now if the peak moves, that's going to impose directional selection on one or more traits. There's not a one-to-one correspondence between the strength of directional selection and peak movement, right? And that's something you need to keep in mind. So the peak movement parameter that describes peak movement is not necessarily going to be beta, right? Beta is going to be a function of where the population bivariate mean is relative to the optimum, right? And depending on the nature of the system, beta and the position of the optimum or the movement of the optimum could be quite different from one another, right? So just keep that in mind. But anyway, let's ask the question, well, what happens if we just have the optimum move gradually over time through phenotypic space? Well, then the population is going to evolve to track that optimum, right? Because selection is going to cause the population to follow the optimum, the population mean. What happens to the G matrix? That's the question. All right, so what happens is even without mutational or selectional correlations, peak movement by itself, so the movement of the bivariate optimum, actually stabilizes the orientation of the G matrix. And what happens is it actually stretches out in the direction of peak movement, right? So the optimum moves, you have a G matrix, say it's round to begin with, it stretches out and follows along behind the optimum, right? Because it wants to come home to its optimum. It's pretty weird. All right, so that's one result. So another result is that if you already have an eccentric G matrix, okay, and now the optimum moves, if the optimum moves along the long axis of the G matrix, it makes the G matrix more stable, right? If the optimum moves across the long axis of the G matrix, it destabilizes the G matrix a little bit, right? And the way to think about it is the rounder the G matrix gets, the less stable it gets. Directional selection stretches the G matrix in the direction of directional selection. And so if it's stretching it to make it more eccentric, then it stabilizes it. If it stretches it to make it rounder, then it destabilizes it. So it's actually pretty simple conceptually. Another interesting result is that if you have a moving optimum, so now just say that the optimum is moving in the direction of trait 1 phenotypic space, right? So we're moving the optimum for trait 1 and leaving the optimum for trait 2 in the same place, right? So we're really imposing selection on trait 1, right? And not changing the optimum for trait 2. Does that make sense? Okay, so what we see under those circumstances is that obviously the population mean is going to follow along behind the optimum. But if you have a strong genetic correlation, what you'll find is that actually trait 2 then moves away from its optimum. So even though it started out at its optimum, the correlated response to selection, the trait 2 optimum isn't moving, right? It stays in the same place the whole time. But because there's selection on trait 1 now, so here's the optimum for this G matrix, and this is the optimum for that G matrix, and so on. So this is over time. These are 200 generation intervals or something like that. Well, what happens is as that optimum moves, trait 1 now tries to evolve to keep up with the optimum, but it actually causes trait 2 to become maladapted due to a persistent correlated response to selection in trait 2. So just moving the optimum for trait 1 can cause trait 2 to become maladapted if you have a genetic correlation between the two traits, right? And so now, even though only optimum 1, the optimum for trait 1 is moving, you're going to have directional selection on both traits now because they're both displaced from their optimum. But the displacement for trait 2 is just due to a correlated response to selection, right? So that's an interesting result that I think is actually pretty important when we start thinking about things like climate change. All right, and then I'll just skip that one. All right, what about reconstructing net beta? Well, it turns out that we addressed this in one of our papers, and if you know the average G matrix over the entire run, just the average, all you need to know is the average, and you get a pretty accurate reconstruction of net beta. If you only know the contemporary G matrix, which would be the G matrix at the very end of the run, you can still kind of reconstruct net beta, but there's a lot of noise under some circumstances, right? So the reconstruction of net beta should be taken with a little bit of a grain of salt. All right, so the last thing I want to talk about with respect to G matrix stability is what about more realistic models of the movement of the optimum, right? So if we have episodic or stochastic movement of the optimum. So that previous section, the optimum just moved very smoothly every single generation, a very small amount. What if it takes big jumps like this? Or what if there's stochasticity so that it kind of goes all over the place like this as it's moving with a net directionality but with some stochasticity built in there? All right. And so the effects of that, so this is what it looks like for a steadily moving optimum. So this is the size of the G matrix. It's just the sum of the variances, so the total genetic variance in the system. This is the eccentricity, where lower values are actually more eccentric. This is the angle, the thing that we were talking about mostly. And then this is just beta, the strength of selection on trait 1, just to give you an idea of what happens with a steadily moving optimum. Right? So just remember, you don't need to worry about what these values are or anything. Just look at the way this picture looks. And then we're going to compare it to an episodically moving optimum in the next slide. So for the most part, things stay the same. But there is clearly an effect of an episodically moving optimum. Right? So you can see up here, the effect is most noticeable with respect to the size of the G matrix, the total genetic variance. And what you get are these cyclical changes in the genetic variance, the additive genetic variance, due to that episodically moving optimum. That corresponds with these spikes in beta, the strength of directional selection, every time the optimum moves, right? So the optimum moves, beta is very, very strong right when it jumps. And then gradually as the mean moves closer to that new optimum, beta gets weaker because now it catches up with the optimum. And then it jumps again, so you get strong beta again, and then it gradually catches up, and so on and so forth. And so what we see, this just compares the steady case to the episodic case. We see that the genetic variances show this cyclical pattern of increases and decreases in genetic variance. The increase occurs right after the optimum jumps. And there's very, very strong directional selection that results in a large increase in genetic variance. And then gradually it goes back to a lower level once it catches up with the optimum. And this is a case where only the trait 1 optimum is moving, but you also see an effect on trait 2, the genetic variance in trait 2, because all the loci are pleiotropic, right? So whenever the variance in trait 1 changes, that also has effects on trait 2. In contrast, for a steadily moving optimum, you just kind of reach this equilibrium level of variation, and it just kind of stays at that forever. And beta stays about the same because you're almost always the same distance behind the optimum.
What's the graphical convention? Is that a standard deviation or a standard error of runs on either side? Is that what that is?
Yeah, it's some kind of, I think it's a confidence interval. Yeah. Like a 95% confidence interval. Yeah. All right. As far as stochastic movement of the peak, you can kind of see the effects here. So stochastic movement, if you add stochasticity, then you get the same basic pattern, right? So each one of these sections, everything else is still true, but now we're adding a couple of different wrinkles. You get the same basic pattern, but the effects are dampened a little bit by the stochasticity, right? So here's an example just showing the effects of the direction of peak movement on G-matrix stability. So here we just have a static optimum, and here's what the G-matrix looked like under the static optimum. And then we compare that under the same parameter combinations, but when the optimum is either moving up and to the right, or down and to the right, right? And you can see the G-matrix gets bigger, and it becomes oriented with the direction of peak movement, right? Does that make sense? Well, if you add stochasticity in, then the same thing happens, but it's not quite as clean, right? It doesn't quite orient exactly in the direction of peak movement. And the reason is that the peak isn't always moving the same direction. Sometimes it's moving at some unexpected oddball angle compared to the normal direction of peak movement. The same thing happens. Remember, I told you that if we have correlational selection and mutational correlations that are aligned, then we get stronger peaks or stronger orientation of the G matrix. If you add stochasticity, then those effects are dampened a little bit, right? That's all that really happens, right? So if you have very high stability conferring parameter combinations, then stochasticity actually decreases the stability a little bit. Or if you have combinations that are really bad for G-matrix stability, then the stochasticity increases the stability a little bit. That's pretty much all it does. Another interesting result, though, is that if you add stochasticity to the movement of the optimum into the model, it actually increases the risk of population extinction, right? So compared to a steadily moving optimum where the G-matrix kind of stretches out and can follow along behind that steadily moving optimum, if now you have random jumps in unexpected directions, what we saw is that we were much more likely to have population extinction in those runs with stochasticity than those without. And the more stochasticity there is, the higher the risk of population extinction, right? And the reason is that you may have a G-matrix that's really conducive to evolving in a particular direction, then suddenly the optimum moves in some unexpected direction, and that causes a huge population decline. All right, so the conclusions under a moving optimum are that under an, oh, you have a question?
If I could just, you were talking about extinction. Are those then just extremely rapid extinction events? Because at the beginning, when you were talking about setting the model, you put some fairly specific constraints on population size.
Yeah, so in this model, we stop to run if it drops below. So technically, we're not modeling extinction per se,
Right.
but if it drops below the carrying capacity, then we consider it on the road to extinction. This has been, sort of supported by other studies that I've done.
Okay.
But basically, if it drops below the carrying capacity, we terminate the run. And so really, we're talking about runs that had to be terminated due to an inability to maintain a carrying capacity. Yeah. All right, so these are kind of the conclusions from this section. So the dynamics of the G-matrix under an episodically or stochastically moving optimum are similar to those under a smoothly moving optimum. The usual things, strong correlational selection and mutational correlations promote stability. Movement of the optimum along genetic lines of least resistance promotes stability. If everything's aligned, then you see a more stable G-matrix. Just movement of the bivariate optimum provides some G-matrix stability. And stochastic and episodic models of peak movement increase the risk of dropping below the carrying capacity at least, which increases your risk of population extinction.
So if you have the steady movement and the optimum, when it starts moving, does the G-matrix sort of start to stretch out and then get more and more stretched out as it continues moving? And then if the optimum stops moving, does it become more circular again? Or does it stay in that stretched out?
So it stretches out, but it doesn't continue to keep stretching out. It stretches out to sort of an equilibrium, sort of quasi-equilibrium shape, and it'll maintain that for a very long period of time. And then if the optimum stops, it'll go back to being round. All right, and then another question.
You're making it
terminology is making it sound as if G-matrix instability is this catastrophic situation. But as you've pointed out, if the G-matrix is nearly circular, then the axis gets very unstable, but the actual covariance matrix doesn't
hardly
change
changes
that much.
Yeah.
I mean, you take a circular matrix and just make it a little bit wider or a little bit
Yeah,
taller. That can flip the axis, but anybody looking at those two matrices would say, wow, they're pretty similar.
Yeah, that's exactly right.
So should we care that much about G-matrix stability in that case?
Yeah. Definitely.
Well, we all have our tastes.
No, you're right. If it's round, then it just gets crazy unstable, but it doesn't really make any difference. Yeah.
You showed a slide where when during the episodic change, a big jump, the genetic variance increased immediately after the new optimum.
Right.
And then it like gradually decreased. So I'm trying to think like what causes that increase immediately? Is it are the breeding values still normally distributed, or is there some sort of like bimodal distribution there?
There isn't a bimodal distribution, but the cause of it seems to be rare beneficial mutations that are sweeping to fixation, right? So as those increase in frequency in the population, it increases the genetic variance, and they continually do that, right? So they're arising and sweeping to fixation. That seems to be the cause. The directional selection does cause skewness in the phenotypic distribution, and that's another effect. And so actually the response to selection is not exactly predicted by the Breeder's equation, because if you want an exact prediction, you have to take into account the fact that directional selection actually causes skewness in the phenotypic distribution, and that also affects the response to selection. So you need to take into account higher moments of the phenotypic distribution. And that can be a fairly large factor. I mean, that can be a factor of 10 or 20% that you're off. And so you're not going to get an exact, because directional selection causes skewness in the phenotypic distribution, and you're measuring directional selection is the thing you want to measure, but you don't take into account that factor, you're always going to be off by some amount, unless selection is very weak.
But the skewness, would that artificially inflate the variance at all? Or?
That might contribute a little bit to that increase in variance. Yeah.
Thank you.
I'm wondering also about the assumption that you've made, which is that the effect of mutations has an average zero effect. That is, it's going up or down with an average effect of the mutation of zero. In cases where a phenotype is reaching a limit, say some sort of developmental limit or maybe functional limit, you could have a situation where most mutations would tend to push you away from that upper limit. Could you comment on how that might influence this picture?
Well, I mean, that would have a dramatic effect, I think. I mean, it depends on the magnitude of the bias and mutation, but it would certainly, I mean, that by itself would drive change in the population mean fairly strongly.
I mean, it would mean that.
But if it's opposed by selection, I don't know what it would do. Yeah, that's a good question.
Selection might be pushing you up against a limit, which the structure of your organism was just going to have a hard time getting all the way to that limit. As you get up near it, most of the mutations would be pushing down away from it. The mean mutation would be moving back away from it.
Yeah.
And so you'd sort of, I think you'd stall out close to the limit and just have mutation mostly putting in deleterious mutations in that case.
Yeah, and I don't know, as far as like what that would do to the structure of the G matrix, I don't know. Yeah, that's a good question. We've never even looked at that, so I don't know.
Yeah, okay, this is on. I have a question that I don't quite know how to formulate. I think part of it is because I don't think I fully understand all this talk. But so earlier today we were talking about G matrices changing over time. The question was, how do you describe that mathematically? And you pointed out some paper involving tensors that's probably hard to read. So the way I'm thinking about this is each G matrix corresponds to eigenvectors. And so if you have any two G matrices, then there is a unique linear transformation, a unique matrix that takes one basis to the other basis. So that's just a matrix. So the evolution of a G matrix over time, the way I see it anyways, is just it's a matrix valued function. And it looks like you're, and so I was just wondering, it's not quite in the scope of this talk, but is this a framework or an approach that you've thought about or people have done? Because here I see the, you're simulating
No,
it.
I hear
I'm
what you're
wondering
saying.
if you can fit the results of the simulation to a matrix value function. I don't know if that's clear, but if you have two G matrices, like, so think, I'm going to do 3D instead of 2D. This thing, so here's your eigenvectors, and there's another eigenvectors. Those differ by a change of basis matrix.
Yeah.
And and so all you're doing as this thing is evolving over time, there is a function. I'm going to call it A of t,
Right.
capital A of t, which is a matrix valued function, which is describing exactly what's going on. Now there's stochasticity, but can be thought about modeling that and looking and trying to fit it to something like that.
Yeah. No, that might be useful. I don't know.
Yeah, it seems easier than that tensor thing you were talking about earlier.
Yeah. Maybe.
But in the case where the variance in the two eigenvectors is about equal, so the matrix is basically circular.
Right.
You again, that kind of description would seem to suggest very dramatic differences between two matrices that are almost really the same. It's just that their eigenvectors have spun, but actually they're nearly circular.
Right. Well, I mean, there's a difference of the actual model and the stability of the model, I think.
It's just that it would lead, I mean, I'm not sure I understand your model, but it would lead to describing large changes where the matrix itself the covariance matrix that you end up with might be almost the same.
Have to think about that. But
Yeah, I mean, if you think of two nearly circular covariance matrices, just stretched slightly more in one direction, stretched slightly more in the other.
so just like this.
Yeah, then the eigenvectors can flip a lot, but the matrices are nearly the same. The covariances are nearly the same.
Well, if they're slightly rotated, then the eigenvectors haven't changed much at all, right?
No, but if I give you a circular matrix that's stretched a little bit north-south, and another circular matrix, but not quite circular, stretched a little bit east-west,
Okay.
the main eigenvector has flipped 90 degrees, which is a very dramatic change, except if you look at those two covariances,
see
they're
what you
nearly
mean.
the same.
Yeah. I'm sure there's a way to get around that. I think so, but I'll think about that.
So I hear what it's Tyler back there, isn't it?
No.
Well, anyway, so who's ever back there? So, but I am back. Oh, yeah, okay. I couldn't. It's right directly behind me, so it's straining my neck. Anyway, who's ever back there? So you'd also want to try your function approach to the eigenvalues, right? Because the whole thing isn't just the change in in the one angle of the eigenvector. So in general there are three parameters. There's either two variances and a covariance, or an eigenvector angle and two eigenvalues, an eigenvector angle and a two. And so you probably need a function you need three functions to explain everything that's going on, I think, with what you're talking about. If I understand.
Matrix value function that takes everything into account.
Oh, it does?
Yes.
All three parameters?
Yes. Because there's three.
Okay, so I like it even better, even more.
But isn't the correlation like the tangent of the angle? So in the case that Joe Felsenson was mentioning, if you measure instead of like cardinal or coordinates, right? You measure polar coordinates and you measure the angle, then you notice that switch or 90 degrees, right?
Yeah, but that-
and that way you could distinguish like this nearly circular case that has rotated completely 90 degrees instead of modeling the whole matrix, but modeling the angle
Right, but
determined by
that's
the correlation.
going to have the same problem that Joe was just describing.
Yeah, I mean that. Yeah, it's a dramatic change. Only it's not really important
Yeah, it has no
because it's
real
two
biological
almost circular
significance.
matrices. They're nearly the same, no matter which way those eigenvectors are pointing. So it's more a question of not whether this is a good formal description of what's going on, but whether some of the large numerical changes you see are really of any interest.
But I think the thing to remember is that so maybe the way to think of it is what I'm calling a stable G matrix is also an eccentric G matrix, right? Because those two things are correlated. And so what we're asking is, when do you get eccentric G matrices? Those tend to be stable, and those provide evolutionary constraints, right? And so in a way, when I say it's unstable, I'm not saying that means that the direction of the response to selection is going to be unstable. I'm just saying that you have a less eccentric G matrix, which doesn't really provide any evolutionary constraints. And so I think maybe we should have focused on, instead of the angle of it, we should have focused on something like the eccentricity more. But the bottom line is that it doesn't really matter because those are kind of the same thing. And when we embarked on this stuff, we didn't really know. I mean, we didn't know what to look at. So, all right. Are there any more questions about that? No? Okay. All right. So the last few conclusions on episodically moving optimums. Episodic movement causes these cycles of changes in the additive genetic variance, the eccentricity, and the stability of the angle. Like I said, stochastic movement tempers stabilizing and destabilizing effects of the direction of peak movement on the G matrix. Stochastic movement actually increases the genetic variance relative to a steadily or episodically moving optimum. I didn't say that before, but that's one result. Selection, this came up as an answer to a question, and I added it to the slide while you guys were talking about it. Selection skews the phenotypic distribution in a way that increases lag compared to expectations, assuming a Gaussian distribution of breeding values. And so you end up underestimating net beta in general due to that skewness. And there are lots of other interesting questions that we can talk about. All right. So are there any questions about that? I'm way over time, but I used a lot of my discussion time during that. So are there any questions or things you want to say about that right now? I'm going to say a few words about epistasis and how that can affect all this stuff, even though you wish I wouldn't.
Continue to learn about what we discovered whenever you talk about this. But so in point 10, you kept saying that selection skews the phenotypic distribution, but I always thought you were talking about the distribution of genetic values. Is that what
too?
Is
Yeah.
that what you meant to say? Because see, look at the way you've got your sentence. It skews the phenotypic distribution, blah blah blah.
And the
compared
genetic
to
values. I mean, in our model, you have to have skewed genetic values.
It does both. Because
Yeah. All right, so basically, the bottom line is that if everything's aligned, it's stable. If there's directional selection, that stretches it out in that direction. And it's only stable when it's eccentric. Does that make any sense? Right, so anything that can produce a strong genetic correlation stabilizes the G matrix. And mutation and correlational selection are the main things. Okay, is everybody on board with that? All right, so the last part that I want to talk about is about epistasis, and there's a reason to study epistasis. So remember at the beginning I said that the M matrix was really important, so the distribution of these mutations entering populations happens to be really important. And so that led us to wonder, well, what determines the M matrix? So how do you get a particular distribution of mutational effects in a particular population? And could selection play a role in shaping the distribution of mutational effects? And so we wanted to address that with additional work. Okay. And so the reason that we wanted to model epistasis is that epistasis actually provides a way in which the M matrix can evolve, right? So epistatic effects can change the allelic effects of new mutations that enter the population depending on the genetic background, right? So epistasis, obviously, it's interaction between different loci. If you have particular types of interactions, then that will change the effects of new mutations entering the population. So basically, we're talking about how the genetic background can affect the allelic effects of new mutations. Does that make sense? Okay, so just think about it as the genetic background. All right, and then the question is, what evolutionary phenomena shape the mutational architecture? And a model involving epistasis provides a more realistic way for the M matrix to evolve than other more contrived ways, probably. I mean, we don't really know because we don't understand epistasis well enough for complex traits. All right, so the question is, what changes that M matrix, right? And we're going to allow, evolution in these simulated populations and see if adding epistasis affects the M matrix. All right, so the model that we used is the multilinear model. And so basically, it's not that complicated. It's only it's more complicated to interpret than it is to set it up. So in the additive model, the phenotype is just determined by the sum of allelic effects across loci, right? So it's strictly additive plus an environmental effect for each individual. In the multilinear model now, it's the same exact model, but now you have an interaction term for each interaction, each pairwise interaction for every pair of loci in your model. Right? So you could have a whole bunch of these interaction terms. And then each one of those has an epistatic coefficient that determines the magnitude of that interaction. You could also have higher order epistasis with higher order interactions, which would have like A1, A2, A3, for example, multiplied together. Right? So basically you just multiply these together and add them to the existing additive effects. Okay, so actually implementing the model is pretty easy. So going from the additive model to the multivariate, the multilinear model is not that huge of a change in the model. All right, and so when we add that, what do we find? So we find a couple of things. So one question is, does it and I'm just going to give some, bare bones results. I'm not going to go on and on about this. I just want to give you just the main interesting results, and just to get you thinking about it. So one thing that happens is that epistasis does increase the genetic, all else being equal, adding epistatic effects increases the genetic variance, which is not a surprise at all. So this is the total genetic variance here, and you can see it's increasing as the values of those epistatic parameters get larger. If they're 0, then there's no epistasis, right? Because every one of those terms is multiplied by 0. There are many of these epistatic parameters, right? There could be, depending on the number of loci, there could be hundreds of these epistatic parameters. So we just draw them from a normal distribution with a mean of 0 and a specified variance, right? So some are positive and some are negative. And if this variance is larger, then the epistatic effects are bigger. The absolute value of the epistatic effects is bigger. All right, so this is stronger epistasis as we move down here. And so what's happening is the total genetic variance is increasing, but the percent that's additive decreases, which you might expect, right? So now you have epistatic genetic variance instead of just purely additive genetic variance, right? So that's not that surprising. One slightly surprising result, though, is that if you look at the total genetic correlation, right? So you're looking at the total genetic variance, there's actually a decrease in the total genetic correlation as you add epistasis, but there can actually be an increase in the additive genetic correlation. So you're actually getting a stronger additive genetic correlation under epistasis than you did without epistasis. But the most interesting thing in this model is how it affects the mutational variances and covariances, so that M matrix, or this is the thing that we were most interested in. And so what we see in this model is actually that it does allow selection to shape the distribution of new mutations. And in particular, what we see is as the population size increases, mutational variances actually decrease. They evolve to a smaller value, right? So the effects of new mutations entering a population are smaller in larger populations. And we think this is because what happens in a large population is the population mean stays very, very close to the optimum. And when you're very, very close to the optimum, there's selection to reduce the mutational variances because they're individuals that produce mutants that are far from the optimum. Those mutants have low fitness, right? So it's weak selection, but it's still, the selection is there nevertheless to decrease mutational variances when the population is very close to the optimum. For smaller population size, genetic drift causes deviations from the optimum, and that decreases the strength of that selection to reduce the mutational variances. Right, so there's selection for smaller mutations or mutations of smaller absolute effect in larger populations. The other interesting result is that epistasis actually allows, when we have correlational selection, epistasis allows the evolution of a positive mutational correlation, right? So you get a genetic architecture that evolves in the population that tends to produce mutations that are positively genetic correlated if there's a positive, if there's positive correlational selection, right? So what's happening is the M matrix evolves somewhat weakly. Selection on it is very weak because mutations don't occur that often, but it evolves to become oriented with the selection surface, right? So now you have a mutational architecture that's compatible with that selection surface that's you know, affecting that's acting on the phenotypes. All right, so this graph shows some of the results. So this is the additive genetic variance as a function of population size. So in a purely additive model, we see this result where the additive genetic variance increases with larger population size. But in a model with epistasis, we actually see a decrease in the standing genetic variation with increasing population size, and that's due to that evolution of those smaller mutational variants, right? So mutations are having smaller effects in these large populations, and so you get less total genetic variance. This is showing that result. So the mutation, this is the mutational variance as a function of population size, and you can see it evolves to be relatively small compared to the smaller populations. So large populations evolve small mutational variances, and those small mutational variances don't introduce as much variation to the population, so you get less standing genetic variance. All right, and then these figures show the orientation of these three matrices, right? So now I've drawn ellipses that are proportional to the selection surface, the G matrix, and the M matrix. And these are from actual data, so if there's no correlation, but I've imposed selection that causes you know, a ridge-shaped selection surface, then the G matrix orients with that, and the M matrix is oriented with the G matrix. What we can do is we can rotate that selection surface in phenotypic space, and you see that, and these are actual results from the model. These aren't just made up, you know, ellipses. These are the results that we saw. The M matrix never becomes that elliptical, you know, it never becomes super eccentric, but it rotates right along with the G matrix and the selection surface, you know, wherever you rotate it, they just stay aligned. So you get this situation where you have triple alignment, and then the mutational matrix reinforces those genetic correlations somewhat. It's not a really strong effect, but it's definitely happening. All right. So that's pretty much all I wanted to say about epistasis, but epistasis allows the mutational architecture to evolve, and when we allow that, natural selection actually shapes that mutational architecture. There's still a lot that we need to figure out about this model, right? So we have some results, but there's still a lot to learn. Smaller populations evolve larger mutational variances. Smaller populations harbor more genetic variance than larger populations in this model, so it's a pretty substantial departure from the strictly additive model. And then we see this triple alignment. The selection surface G matrix and M matrix evolve to be in near perfect alignment. Obviously, we need more empirical data to sort of start to, you know, ground truth some of these assumptions that we've made with respect to epistasis and mutation rates and things like that. But is promising, and it does suggest that natural selection is somehow affecting the mutational architecture of these traits. All right, there are a few implications of epistasis when we start thinking about quantitative genetics. So one is that high apparent additive genetic variation or high actual additive genetic variance doesn't necessarily imply that the underlying genetic architecture involves only independent additive loci. There's a tendency for people to go, oh, there's a really high heritability, so this trait must be composed of many independent additive loci. But in fact, much of the additive genetic variance in these models is arising from those epistatic interactions, right? And it is additive genetic variance, but it's coming from those epistatic terms. So the interaction terms are causing additive genetic variance. Mutational variances and covariances can be shaped by selection when epistasis enters the picture. For traits affected by large numbers of genes, there could be sort of a mind-bogglingly huge number of these epistatic coefficients if epistasis works anything like this multilinear model. And if that's true, then it could be very, very difficult to find these interaction effects in actual genome-wide association studies or QTL mapping studies. And then I think this causes us to think a little bit differently about the mutational architecture, because at least I thought about it as like the mutational architecture is like this developmental thing that's sort of separate from natural selection, and it's the raw material of evolution, and then natural selection can act on that. But what we're seeing when we start thinking about epistasis is actually there's feedback from natural selection that affects then the mutational architecture, and then that changes the nature of how natural selection can act on the population, and then in turn that changes the mutational architecture. So there's a very complex relationship between the mutational architecture and natural selection. All right, and so that's the last thing I want to say. I think there's a lot that we can learn, though, still about epistasis, and it's very important. But all right, so thanks for listening to me, and I guess we have a few minutes for questions or discussion.
I think we should give a round of applause.
I just have a technical terminological issue. You're talking about these epistatic effects, which are going to, in the usual variance breakdown, they're going to be additive by additive effects, I believe. And you're calling them additive. So
no, I think
when you say the additive genetic variants, are you technically?
No, I think they're actually the additive genetic variants. So not all of the epistatic terms.
Oh, I see. So when you fit the kind of Fisherian terms, some of it comes from that.
Yeah, exactly. Some of it's specifically what he would call additive genetic variance. And it depends on all kinds of things like linkage disequilibrium and allele frequencies and things like that, how much of it's additive versus additive by additive. But yeah, they're making a direct contribution to additive genetic variance in the Fisherian sense.
I'm just curious, so a lot of the discussion was about the shape and the stability of the G matrix, but is it important or useful to try to understand how quickly or rapidly it moves towards the optimum or the peak?
Yeah, I mean that's of interest too. Yeah, and that's a sort of a separate issue. I mean that's sort of just figuring out what's the response to selection going to be, and that's relatively straightforward. Once you know what the G matrix is, and the strength of selection and the position of the optimum, you can almost perfectly calculate, how fast it's going to keep up with that. And so that's certainly important, but it's sort of separate from what we were talking about, which is what affects the genetic architecture and mutational architecture. But yeah, that's what we want to know. That's why we want to know it, right? Is so we can figure out how it keeps up with the optimum, or how selective breeding would affect things, or how an environmental change would affect, the phenotype, all those sorts of issues. Yeah.
Okay, this is kind of a simple mind, but I just don't really understand. I mean, do you have any, I know that the result in which epistasis led to additive genetic variance came out of a simulation model, but like functionally, how do you think that would happen?
How
I'm just not
an
understanding.
epistatic
Yeah,
interaction. It's because there are shared epistatic effects between parents and offspring. I mean, so some of that. So depending on the genetic architecture of the traits, some of those epistatic effects contribute to parent-offspring resemblance, and so they end up going into the additive genetic variance. I'm not going to be able to explain it in a way that makes
Well,
any sense.
because I can picture that, but I can picture the reverse too, and so I don't know. I just need something for my brain to stick
Yeah,
to
so
make it make sense
they're
to
not
me.
easily separable. So
Yeah.
those epistatic coefficients aren't easily separable from the main effects of the alleles, and so you can't just calculate. So you cannot simply calculate the additive genetic variance in that model by looking at the additive effects of the alleles. You won't get anything close to the additive genetic variance. You have to come up with these gigantic equations that take into account allele frequencies and all those epistatic interactions as well, and their contribution to the additive genetic variance. And so the point is just that those, the only way I can say it that would make any sense is that those epistatic terms, some proportion of those values is contributing to what we call the additive genetic variance. And it's just additive genetic variance. It behaves exactly the same way. It causes parent-offspring resemblance and all those sorts of things.
So, sorry, can I take a stab at kind of explaining what maybe you were asking? And this may be way off, and it's off. You can just tell her to disregard what I say. But so in the beginning, when Joe Felsenstein was talking about the additive effects being a slope through 3 points, right? So, in a particular genotypic background, a mutation or, genotypes might have a certain pattern. But in a different population where there's, say, a different genetic background, those 3 points could assume different relative positions, and therefore the slope through those lines would be different depending on the genetic background of the population, right? So, that's what epistasis is, right? And so the additive genetic variance due to that one locus will change because the relative position of those 3 genotypes is different depending on the suite of genetic backgrounds in the population. So you could think of it as I'll use like coat color in dogs or something like that, right? There's the white, the brown, and the black, right? Only in a certain genetic background can you see the black and brown differences, right? So in a population where you have that mutation, you see that genetic variance. But in a population that is fixed for, say, the mutation that affects downstream that allows any coat color production. So in that population, if everybody has the yellow lab allele, there can be no variance in brown and black because it's fixed for the yellow. And so that's how epistasis functionally can play into additive genetic variance.
To say pretty much the same sort of thing, that with the epistasis terms, just even when there's just 2 loci, one locus changing its value can cause the other locus, the values of those 3 things to get steeper. So if you don't have any epistasis, you can't do that. Then each locus will just make the different genotypes will make the same contribution no matter what the other genotypes are. Put these epistasis terms in, and one locus changing frequency can cause the other one to suddenly have more distinct values or less distinct values, and that's what's going on here. And I think it's not that with all these epistatic terms that they're having random effects. It's that natural selection is sorting out, is biasing you in certain directions towards more or less additive genetic variance by moving gene frequencies so that some of those terms are starting to have a big effect and others maybe not so much.
Yeah. That's definitely happening. So you see a situation where, like in those large populations where we see a reduction in mutational variances. What's happening is loci with large negative epistatic coefficients tend to evolve larger positive effects so that they sort of cancel each other out. And so you end up getting the allelic effects can evolve. The epistatic coefficients in that model are parameters. They never change during the run. And so changes happen by the evolution of those main effects. And so you get favorable combinations of epistatic parameters with main effects that actually reduce the effects of new mutations by sort of canceling out the effects.
I'm just thinking about empirical data, and I can't recall it very well, but doesn't it seem to be a rather positive correlation when you look at populations of varying size, the positive correlation between the population size and the amount of additive genetic variation that's estimated in them? In empirical data?
Yeah, I don't know. Yeah, I haven't looked at that actually in any kind of detail. That's what I would think. Yeah, but I don't really know. I mean, there's definitely a positive correlation between molecular variation and population size, or kind of. But yeah, I don't know.
Because
I haven't.
That would be a way of testing, because what your model would predict is the opposite, right? Or at least to some extent, the negative correlation between population size and additive genetic variation.
Yeah, if we're in the ballpark, if our parameter values are in the ballpark of those populations, yeah, that would be the prediction. But yeah, I haven't tried to look at that. That's a good idea. That's something that's worth looking at. Yeah.
The results definitely flying in the face of intuition.
Yeah, to some degree.
I have a question. I hope you don't mind me asking again. In the beginning of this lecture, you said something that sounds really mind-bending to me. So you said when there is a pleiotropic effect, it doesn't necessarily mean that there's genetic covariance between the two traits. Yeah. And I was hoping throughout this lecture I will understand that more, but it seems I'm just as confused. So I wonder if you can comment on that.
The reason is that in the absence of any mechanism causing allelic effects to be correlated, even though you have pleiotropy, the positive allelic effects are just as often associated with negative allelic effects as they are with other positive allelic effects. And so you get no genetic correlation because they just kind of cancel out. And so even though each allele has effects on both traits, it's equally often positive and negative as positive and positive, or positive and anything else, unless you have some mechanism that's getting rid of some of those combinations of allelic effects. And then as soon as you do that, then pleiotropy helps facilitate the maintenance of a genetic correlation. It's much more effective than, say, linkage disequilibrium would be by itself. But you have to have something to get rid of some combinations of alleles, like the negatives with the negatives, or you don't get a genetic correlation.
I guess maybe I'm still a little unclear as to exactly what's meant by that. So the way I'm thinking of it, a particular instance of an allele, of a pleiotropic allele, existing in a population, with its specific positive or negative effects, that allele will generate a genetic correlation, I would think. But your general framework for assuming a bivariate normal distribution of mutations, and they are pleiotropic, but they have no bivariate, correlation. That mutational scheme will not generate genetic correlation, but a particular instance, a particular pleiotropic allele will generate genetic correlation.
That's true. If you had many individuals that had that allele, then you would get a genetic correlation. But that too would erode. So if you had a mutational correlation of 0, say, so you just have a bivariate normal distribution with no correlation, for the new allelic effects, that correlation would also erode over time, because what would happen is that combination of alleles due to new mutations would eventually get eroded by those new mutations to the point where you just have random effects at the two traits. But yeah, you're right. If many individuals have that same combination, that same allele, then that could contribute to a genetic correlation.
So one of your results I think was interesting is the smaller populations evolve larger mutational variants. If you go to the extreme case where you have a very, very low population size, I mean, does it matter? Like the actual mutational target is so small and you have two individuals and you have a mutation rate of 0.002, chances are the next generation is not going to have a mutation.
Yeah, I think in the smallest populations, I think you would not see this effect, because I think it's driven by selection, very weak selection on the mutational correlation, and selection would be so weak that I think you wouldn't get any kind of, if you had a population size of 2 or something,
Right.
I think the effects of drift would override any kind of effect that you would get. But I don't know. Our model would crash at that population size, I think. So I haven't tried that.
So the mutational variation that you get when you had 128 individuals, is that bias anyway? Like if you just pulled 128 random samples, would you have a larger variance than if you pulled from the same distribution, pulled 1,000 random draws? You would have a better estimate, maybe a tighter variance, or would it
Of
be
the
biased?
mutations, you mean?
Yes, I guess this is more of a probability theory question.
So you're asking whether our estimation of the mutational variance
how
is
that
different in those populations?
Yeah.
Yeah, so the way we did it is we're not actually measuring actual mutations that occur, because there aren't enough to do anything with that. So for each time we want to measure the mutational variance in a population, we have to make a copy of the entire population and then induce a bunch of mutations, like 200 mutations per individual, and then average across all the individuals to get the mutational matrix. And so it's an average, and so the estimation of it's exactly the same regardless of population size.
Right.
But yeah, so you can't actually measure anything on the actual mutations because there aren't enough of them any particular generation.
Okay.
Another question.
I was wondering if it was known empirically how common mutational correlations are. I mean, ones that don't just kill the animal dead or whatever,
Yeah, there have only been a few studies that have measured mutational correlations, because you have to do a mutation accumulation study, and then measure multiple traits and mutation accumulation studies. And I don't right off the top of my head know what the results are. I mean, I think mutational correlations are reasonably common, but do you know, Steve?
I'm thinking of studies by Suzanne S.
Yeah. Studies of mutational accumulation, when people do multiple traits, they don't always, but when they do multiple traits, I can't say always, but you do see mutational correlations, covariances. They're there. But
It's a
right
real
off the
thing.
top of my head, I don't know what the distribution is of mutational correlations. And there aren't a lot of estimates. I mean, it's only a handful of studies that have looked at it.
I don't know about the empirical studies, but you would think that when there are different loci acting on a trait through common developmental pathways, or two traits through some common developmental pathway, that could have a very strong effect in pushing pleiotropy in particular directions and pushing interactions in particular directions. That these simulations are not building in any particular mechanism, so they don't get to check that.
Right. Yeah, but I agree that I would expect that